Vivo Biotech Limited is a contract research services company offering its services in the segments of custom research, biologics, non-clinical, and clinical development services. The state-of-the-art preclinical research facility spreads over 125,000 square feet in 5 floors, designed in compliance with ALAC Association for Assessment and Accreditation of Laboratory Animal Care International and GLP Good Laboratory Practice Guidelines with inputs from international consultants and experts. The facility has circular design with optimal and effective space utilization for animal handling, experimentation, and analysis. Sterile and non-sterile areas are clearly demarcated, functionally secured to ensure the compliance of GLP and internal quality assurance requirements. A robust building management system has been implemented for monitoring and managing the eco-parameters, access control, and HVAC parameters. Environmental factors are controlled by BMS such as 12-hour light-dark cycle, temperature and humidity with 100% HEPA filtered air replacement at the rate of 15 to 20 air changes per hour. Noise levels are maintained under permissible limits. Walls and floors are designed to meet international standards. Pressure gradients are maintained among animal holding rooms, procedure rooms, and the return corridor. A separately demarcated test substance control office controls test material as per the test drug handling and accountability procedures. Wash area connects to clean stores through a double-door autoclave. The sterile laboratory where irradiated feed and the purified water are assembled to maintain the biosafety levels in animal holding rooms. Animals from external resources are received in animal receipt rooms separate for rodents and non-rodents. They are transferred to clean cages under laminar air flow and are moved through pass boxes to respective quarantine rooms. All the animals included in studies are confirmed for their strains and health status is evaluated using different biotechnological methods. Scientific staff has clean corridor access procedure comprising of stripping, shower and sterile clothing. Animal holding rooms are specific for species and the study. Handling of animals and experimentation are performed by well-trained personnel in controlled and well-equipped environment as per standard operating procedures and approved protocols. These rooms are connected to the procedure room where administration of test substance, body weight recording and observation are done under controlled environment to maintain the integrity of the study. All the samples from terminal holding room and terminal bleeding room are transferred to bioanalytical lab through pass box, where complete analysis of blood and urine is performed using hematology analyzer, urine analyzer, and biochemical analyzer. The facility houses best of the instruments from reputed vendors for exceptional quality conduct of the studies. The laboratory is secured with restricted access and served by a fully computerized laboratory information management system. The terminal holding rooms have metabolic cages where metabolic parameters are quantified, terminal body weights are recorded, and the body fluids are collected. This room is connected to terminal bleeding room where smears are prepared, blood is collected and processed for further studies in hematology and biochemistry. All the samples from terminal holding room and terminal bleeding room are transferred to bioanalytical lab through passbox, where complete analysis of blood and urine is performed using hematology analyzer, urine analyzer, and biochemical analyzer. Through pass boxes, the euthanasia room connects to the necropsy room, where necropsy is carried out and gross pathology examination, organ collection, weighing and fixing are done. Tissue sampling is done under fume hood and with well-planned labeling, indexing and proper documentation. These sample tissues are passed through pass box to histopathology laboratory, where tissues are processed, sectioned, fixed and stained. We can provide sections as thin as 4 microns. These sections are evaluated and reported by expert pathologists. Biomedical waste is disposed of as per applicable guidelines. Areas of genotoxicity and cytotoxicity studies are demarcated for different cell types where in vitro studies are conducted. Vivo Bio offers in vitro development services for sponsors including ADME studies, cell-based functional assays, toxicity screening assays. We also offer cytotoxicity and genotox tests as part of the regulatory tox battery. We are showcasing a representative workflow here of a case study of Amy's test to identify the mutagenic properties of the drug molecules or chemicals. Driven by validated and standardized operating procedures as per GLP guidelines, trained scientists perform standard assays in cytotox and genotox as per OECD guidelines and develop custom assays as per sponsor requirements. Experienced scientists man the in vitro lab with equipment such as phase contrast inverted microscope, autofocusing trinocular microscope, electrophoresis equipment, gel dock system, HPLC, etc., offering the sponsors advantages of standardized and validated methods, aiding in minimal time for start of studies. We are showcasing preparatory activities for cell-based assays. Vivo Bio focuses on providing contract research services by leveraging the technical intellect of scientific manpower, standards accredited infrastructure, and regulatory guidelines compliant best practices. With plans for adding additional capacity and resources to accommodate fixed bid, custom and FTE engagements, we endeavor to be your preferred partner for early-stage drug development requirements and assure you of exceptional quality study conduct on the strength of our customer-centric processes, validated standard operational procedures, and experienced resources to meet and exceed your expectations on quality, time, and budget.